Okay, now moving further, the next subheading will be construction of recombinant DNA molecule. Okay, just to recall what we have done till now, we have selected our DNA insert, we have isolated our DNA insert, then we have selected a suitable vector which will carry our DNA insert. Okay, so now what we have to do, we have to join these two. We have to construct the recombinant molecule. That means we need to allow the the vector DNA, that is the vector. If we have chosen cosmid as the vector, so we are utilizing the DNA of the cosmid and mixing it, joining it with the DNA insert, thereby making a recombinant DNA molecule. So under this, understand, the DNA insert and the cloning vectors are cleaved using the same restriction enzyme. Why this was being done? Because the DNA insert and the cloning vector should have the sticky ends complementary to each other, so that if we will cleave them using the same restriction enzyme, you remember what was happening, the same restriction enzyme will cut at the same recognition sequence and the same protruding ends will be there. So the protruding ends or the sticky ends which are produced can be joined easily. So both should be cut using the same restriction enzyme. Now, why this is important? Because they must have sticky ends. These sticky ends are mixed and incubated in the presence of DNA ligase. This is the next step. We have taken both the DNAs, the DNA insert, that is the DNA of interest, and the cloning vector DNA. We have mixed them and both have sticky ends. So, of course, they will be sticking together, but not without DNA ligase. So what we are doing? We are incorporating this, incubating them in DNA ligase. DNA ligase you have already studied in Okazaki fragments, remember? So Okazaki fragments are also joined by this DNA ligase. What was the basic technique involved there? DNA ligase was forming the phosphodiester bonds between the small fragments and making them a continuous strand. Yes, same thing will be done here by DNA ligase. DNA ligase is capable enough of forming phosphodiester bonds. So now it will form the phosphodiester bonds between the DNA insert and the cloning vector DNA. Plus, what will happen? The hydrogen bonds will be formed between the complementary nucleotides because it is a sticky end kind of thing. So, sticky ends will form, will be complementary base pairing will happen in between the sticky ends, and this complementary base pairing is nothing but hydrogen bonds. Of course, so hydrogen bonds will be formed. So two types of things are involved here. First, the complementary base pairings, which involves the hydrogen bond formation, and second is the DNA ligase, which is forming the phosphodiester bonds between the sticky ends of the DNA insert and the vector DNA. Clear? So these are the basic steps. A quick revision again. DNA insert and the cloning vector are cleaved using the same restriction enzyme. You know the reason why. This is important as they must have the sticky ends. Next, these sticky ends are mixed and incubated in the presence of DNA ligase. Then, hydrogen bonds are formed between complementary nucleotides of the sticky ends. Then, the DNA ligase forms the phosphodiester bond between the sticky ends of the DNA insert and vector DNA. Okay, so they also have to be joined. The sugar phosphate backbone has also to be joined. So how they are joined? They are joined by the DNA ligase by the formation of phosphodiester bond. Clear? Okay. Now the next step is what we have done till now. We have taken a DNA insert, we have taken the vector, we have mixed them. Now our recombinant molecule, recombinant DNA molecule is ready. Now the next step will be incorporating this recombinant DNA molecule into a host, so that when the host replicates, our DNA also replicates. Okay, so before we incorporate this, what we have to do? We have to select a particular host. What kind of host is required? So moving forward, the concept of non-rDNA. Okay, yes, one more point I want you all to to understand here. Sometimes what will happen? Sometimes the recircularization of sticky ends take place without the incorporation of DNA insert. This produces non-recombinant DNA. Are you understanding this? What we have done, we have used the restriction enzyme to cut the fragment, to cut the whole, the you can say the vector DNA. So sometimes what will happen, the vector DNA will recircularize again without incorporation of the DNA insert. So that will not be a recombinant DNA. You understand? Why it will not be recombinant DNA? Because it is not having the DNA insert. So is it of any use for us? It is of no use. Okay, it's of no use for us. Why? Because what we are doing, the basic aim of this whole technique of genetic engineering is incorporation of the recombinant DNA. But when the recombinant DNA is not formed, that is of no use, the whole technique goes in vain. So what is needed here? Screening should be there, screening should be there, a filtration technique should be there, so that the non recombinant DNA can be uh, left and only the recombinant DNA should be selected. So how it can be done? One possible method should be the DNA insert should also have a selectable marker in them so that they can be identified. This is a very important point. It can come in 1 mark or sometimes in 2 mark. How will you differentiate between the non-recombinant DNA and the recombinant DNA? A recombinant DNA has the incorporated DNA insert also, whereas the non-recombinant DNA do not have the DNA insert, and it is formed because of the recircularization of the sticky ends of the uh, vector DNA only without the incorporation of the DNA insert. Clear? So what we should do? We should have a screening method to segregate these two. And what can be possible screening method? That the DNA insert, here we can, if we have the selectable marker in the vector DNA, that will be of no use because this selectable marker will be present in recombinant DNA also and the non-recombinant DNA also. Clear? So here a selectable marker should be present in the DNA insert of the or the DNA of interest also, so that we can recognize that this particular recombinant DNA has the DNA insert and these do not have. So that makes a very important concept. That is a very important point. You should always remember that there is a possibility of recircularization of the vector DNA without the incorporation of the DNA insert. So the DNA insert should signal us, should give us a signal ki, yes, I am present here. So what can be that possible signal? We it should have a selectable marker like an antibiotic resistant gene present, or it should have any radioactive thing incorporated, so that we can just screen it under the UV rays and find out that whether they are being this is recombinant DNA or non-recombinant DNA. Clear? Okay, moving forward.